Hi, everybody. Okay, here's the second lecture for our remote learning. I finally figured out how to put my face in the corner. Yay. So hopefully this feels a little bit more like we're in class. I had one comment about the audio. Hopefully you're able to hear this okay. I'm using my Bluetooth headphones. If I just use my computer microphone, you can't hear me at all. But if there's ongoing issues with the mic, just let me know. Okay, so one of two lectures this week, we are looking at the Hardy-Weinberg Principle, and like I teased from the last lecture, this is a really important null hypothesis. All right, so we're going to learn what this Hardy-Weinberg Principle is. So as always, we'll start with our agenda. First, I'm going to discuss what assignments are due this week. So first of all, a shout out to those of you who filled out the Google form that I sent around. That was really helpful feedback for me. It sounds like as long as I keep to the schedule of lectures on Monday, really clear expectations for what's due when, due date Sunday evening, that's a schedule people will be able to keep. So let's go with that. So I'll discuss what's due this week. We'll review allele frequencies. So we've been talking about that quite a bit. We're going to dig more into that today. And then we're going to discuss what this Hardy-Weinberg principle is all about. And lastly, we'll do some practice calculations. So we'll go over the goals, what I hope you guys get out of this lesson today. Be able to describe the concept of a gene pool. You may have heard of that before. We're going to calculate genotype frequency and allele frequency. So I have a check mark here because you guys have already done some practice with that. So we're going to continue with those skills. Describe and be able to apply the Hardy-Weinberg principle. So I've broken that down into a couple of things. One, we're going to identify the assumptions that we make when we're using this principle. We'll define these components. So we'll figure out what P, Q, P squared, 2PQ, and Q squared mean. And we're going to be able to use allele frequency to calculate genotype frequency and then go in between those two things. So far we've just been counting. But given a very big population, we'll try to figure out allele and genotype frequency without having to count by hand. So those are the goals, what I hope you get out of this lesson. Okay, so just like I said, we'll dig into what assignments are due this week. So hopefully you have your ignorance book handy. So this is the first assignment, the first reflection. We're going to read the introduction through chapter 3. This is a very short book, so this goes by very quickly. The full document, all the details are on Moodle. Again, this assignment isn't very long. It's not even as long as one of those 2-page assignments you guys have been working on. Just short couple questions about those first few chapters. We will also have a lab 10. So instead of doing what we originally had planned, we have a series of Hardy-Weinberg problems. So a little bit of an extension about what we're going to be talking about in this lecture. So once you've gone through this lecture, you'll have everything you need to know for the lab. This will be set up in a questionnaire just like lab 9 was. So if there's any technical difficulties, please let me know. We can try to iron those out. Otherwise, it'll be just like you did for lab 9. And then everyone should have an email from me with their exam 2, your score, and a key. So just like the first exam, you have the opportunity to correct any questions, and you have the ability to gain 1/4 of the points that you missed. So please get that in by Sunday. All of these assignments, the ignorance reflection, lab 10, and test corrections are due Sunday evening. One thing I forgot to write down here is there will be a lecture quiz after this lecture and a lecture quiz after the second lecture I'll post. Those, again, do any time this week. Get them done by Sunday. So I have Sunday due dates just so that you have the whole week to work on them. But I'm trying to be as flexible as possible, given that we're now spread across all of our homes. So if you require an extension, something comes up, please let me know as soon as you can, and I'm going to be as flexible as I can. But plan on finishing everything by Sunday if you can. So that's this week: reflection, lab, test corrections, the quizzes. And something else that's coming up is your phylogeny presentation. So I've been teasing that I would update it soon. So the document, again, is on Moodle. So for full details of that, please see the document there. Instead of one big presentation, I've decided to put it into two. So instead of trying to upload a big long video, you have two presentations. They should each be 5 minutes. We were going to have another presentation during a lab, but I've decided to just get rid of that. So in order to fulfill that speaking requirement, each video should be 5 minutes. So it's going to be the same exact questions you were answering before, but see that document in Moodle about how that gets broken down into these two. So part one, the important thing is the due date. So part one, your first 5-minute presentation is due next Sunday, April 5th. And then the second one is due at the end of week 14, and that's Sunday, April 19th. Okay, so again, new due dates. The first part, everyone's are due April 5th. Everyone's second presentation is due April 19th. The details are in that document again, but very briefly, you will create a presentation. You will also film yourself giving the presentation. Your slides do not have to be in the video, but you should help me as best you can by referencing specifically which slides you're on. And any questions about that, please let me know. I'm happy to chat about that, figure out different media. Any means you have of recording yourself is fine. Again, if you have specific questions, let me know. And again, first one's due next Sunday. So you have all of this week and all of next week to work on those. That said, now we're going to talk about Hardy-Weinberg. So again, this is a null hypothesis. Very cool idea when we're studying evolution. So for that first goal, we were chatting about what a gene pool is. So for this class, a gene pool is just an idea that if we have a certain population, in this case we have our lovely beetles, that whole population, all of those alleles put together, represent the gene pool. So they're not all physically together in space, but just thinking about all of these alleles together in kind of one mixed bag. So the gene pool, all of the possible alleles of this population are kind of floating around. So when we're thinking about how to calculate genotype and allele frequency, you guys are really good at this already. So let's figure out. So these two right here, these three are our genotype frequency. We have both alleles present. These two down here, big A and little a, are allele frequency. So for genotype frequency, we have 17 individuals. So cross that out. 17 individuals. All we have to do is look for the red homozygous dominant individuals, and there are 6 in this population. So 6 out of 17 individuals are homozygous dominant. When we're looking at heterozygotes, those are our orange beetles. I don't have a good orange color, that's okay. This will also be 6 individuals out of 17. And then our homozygous recessive, our yellow beetles right here, those are 5 out of 17. So we can get that by either counting those 5 or just saying 17 minus 12, which is how many the other two had. Okay, so here are our genotype frequencies. So if you can figure out allele frequency on your own, go ahead and do that while I'm writing this down. But if you need a reminder, we figure out dominant allele frequency by figuring out how many dominant members there are and multiplying it by 2, because we need to take into account that each individual has 2 of these alleles. So we've got 12 of our dominant alleles from our homozygotes and 6 dominant alleles from our heterozygotes. So that would be 18 dominant alleles in our gene pool out of a possible 34, because we've got 17 individuals, each one has 2 alleles. Okay, so 18 out of 34. We do the same thing for our recessive alleles. We have 6 from our heterozygotes and 10 from a homozygous recessive, so that is 16. And we can double check that we have the right answers, because these two numbers should add up to 34. So 18 plus 16 is 34. We are all set. All right, so again, gene pool, just all of these individuals' alleles in one big pool. So our big idea today is Hardy-Weinberg. So that's who these two lovely gentlemen are. In the early 1900s, they were thinking about the implications of Mendel's ideas. They were mathematicians thinking about how you could figure out the probabilities of where alleles are in a population. So this idea is kind of foundational for people that study population genetics. So this Hardy-Weinberg Principle, named after these two scientists, these two mathematicians, is a null hypothesis for studying evolution. So what does that mean? Well, the principle states that in a large, randomly mating population, the genotype frequencies will not change from one generation to the next unless evolutionary processes are taking place. Okay, so we'll get into what large and randomly mating mean, but essentially, we can look at a population. If it's following these rules of Hardy-Weinberg, the genotype frequencies should not change from generation to generation. If they do, and they most likely will, something is going on, and we can attribute that to these evolutionary processes. So that's this idea of a null hypothesis. If our expectations are that nothing will change, our null hypothesis, our Hardy-Weinberg principle, and we see in our results that something has changed, then we know it's due to one of these evolutionary processes. Okay, so one of our goals is looking at the assumptions of Hardy-Weinberg Equilibrium, and this is what they are. So this is a nice graphic summary from the Amoeba sisters. Let's break that down. So for each population, if it plays by Hardy-Weinberg's rules, there will be no selection. So every individual has an equal chance of making it to the next generation. There will also be no genetic drift. So that means that there's not going to be these random changes in alleles. If it meets this Hardy-Weinberg principle, no gene flow. So if we're looking at one population, we're not going to see any incoming information. So when we were playing with those sticky notes and we had them moving from the mainland to the island, we're assuming that that is not taking place if a population is meeting these assumptions. No mutation, right? So we can see our frog over here. We're assuming there's no mutations in our genes. We know based on the previous few lectures that that's always happening at kind of a small rate. But again, this is one of our assumptions. And then we're also assuming random mating. So in a real-world population, lots of things go into who mates with who, proximity, if nothing else. Frogs that are closer together will probably mate rather than ones that are really far away. However, under these Hardy-Weinberg principles, we're assuming that all of these are taken into account. So a population that is in Hardy-Weinberg equilibrium, no selection, no drift, gene flow mutation, and each allele has an equal chance of combining with another allele. So we're in kind of our mathematical space. This doesn't necessarily reflect how populations actually work. So thinking about what random mating means, it assumes that individuals choose their mates at random, no sexual selection, so we didn't really talk about this before, but we're assuming this isn't going on, and no inbreeding. So lots of plants undergo inbreeding if their flowers pollinate themselves. If they're under Hardy-Weinberg equilibrium, that is not the case. Okay, so we're going to connect this idea of populations under Hardy-Weinberg equilibrium with calculating allele and genotype frequencies. So we're going to focus on one gene with two alleles. So all of the problems we're going to do, we're assuming we're looking at just one gene, and each gene has two alleles. We can name those A1 and A2. So usually A1 refers to a dominant allele, and A2 refers to a recessive allele. For our purposes, we're going to define P as the frequency of A1. So one of our goals was identifying these terms. P, the letter P, refers to the frequency of this dominant allele, or this A1 allele, in a population. Q refers to the recessive allele, or A2. So again, P equals frequency. Q equals frequency. So now we can think about how to represent the genotypes. Because we're still working with one gene, two alleles, we know that there are three possibilities. So we either have our homozygous dominant, our heterozygote, or our homozygous recessive. And I've color-coded the alleles just so that we can start to tease them apart. So thinking back to our beetle example, you just notice the colors probably coordinate. So these frequencies can be written in these terms right here. Again, P is the frequency of our dominant allele. Q is the frequency of our recessive allele. So if we're thinking about our gene pool, we can think of just a bag of alleles. And we want to know the probability of pulling out two alleles. So if we think about the probability of pulling out two dominant alleles, we multiply how often we see P times how often we see another P. So the chance that I pull out an A allele, like a dominant A, is multiplied by the chance that I pull out another A. That means I pulled out two in a row. So P squared, P times P, is the probability of an organism having two dominant alleles, meeting all of these Hardy-Weinberg assumptions. So one more time, P squared is the same thing as the probability of getting an organism that is homozygous dominant. The chance of getting two A1 alleles in a row is the chance of getting one times the chance of getting one. So P squared is the probability of getting a homozygous dominant. So same thing with our homozygous recessive. The chance that we reach into this gene pool and grab a recessive allele is Q. The probability of getting two in a row is Q squared. So the probability of getting a heterozygote is the chance of getting a P allele followed by a Q allele, so that's P times Q. But it could also happen that we pull out a Q and then a P. So that's why we multiply it by 2. So another way to think about this, P squared is the probability of getting A1 genotype. 2PQ is the probability of getting a heterozygote genotype. And Q squared is the probability of getting a homozygous recessive, or A2 genotype. If that was hard to follow, there is a really helpful Khan Academy video that I've linked to at the end of this lecture. So bear with me. Rewritten to equal 1, the chance of getting a dominant allele and the chance of getting a recessive allele should equal 1, right? Because we only have these two possibilities. This is just another way of writing our genotype frequencies that we saw on the previous slide. Again, they should equal 1, because those are our only options. So we have a 100% chance of getting one of these three. So if the idea of probability and genotype sounds familiar, it's because we've been using Punnett squares. So Punnett squares are just a way of figuring out the probability of one cross. If we use these terms, we're able to figure out the probability of getting any organism out of this population. So these are very similar. In fact, the person who invented Punnett squares was friends with G. Hardy. Okay, so we'll be able to use these numbers to figure out different things. So in this example, here's our gene pool. We have our A1 allele, in this case it's red, and our A2 allele in blue. So in this gene pool, we have a certain number of alleles, and our chance of reaching in and pulling out a red or dominant allele is 0.7. So 7 out of 10 times I reach in, I'll pull out a red allele. We have a 70% chance of getting A1. We also have a 30% chance of getting allele A2. So here's a way we can write that. P, the frequency of our dominant allele, is the same thing as the chance of getting it, 70%. The chance of getting a recessive allele is 0.3, or 30%. We can use the allele frequencies to calculate how often we would expect to see different genotypes. So if we want to figure out the chance of getting our homozygous dominant, we multiply the chance of getting one dominant allele by itself. So 0.7 times 0.7. To figure out the chance that we would get an organism that's homozygous recessive, we will multiply the probability of getting 1 A2 times the probability of getting 1 A2. So 0.3. If we want to figure out the chance of reaching into our gene pool and pulling out a heterozygote, we just take 2 times the probability of a dominant allele times the probability of a recessive allele. So we'll see how this works on the next page. Okay, so here we are. We're just pulling these different genes out, or different alleles out. So we're reaching in, grabbing some, reaching in, grabbing some. Here's a reminder of our genotype frequencies. Again, this is all assuming that they're meeting those Hardy-Weinberg assumptions. So we can plug in our numbers here. So we know the probability of getting a dominant allele is 0.7. So we're just putting that number in for P. We know the probability of getting a recessive allele is 0.3. So we can plug that in for Q. So 0.7 squared plus 2 times 0.3 times 0.7 and 0.3 squared. So this adds to 1 because we have a 100% chance of getting one of these three. And here are the probabilities of getting each genotype. Okay, so the probability of getting our homozygous dominant is 0.49. So the probability of me reaching into this gene pool and grabbing 2 dominant alleles is almost 50%. The probability of me reaching in and grabbing 1 dominant followed by 1 recessive allele is 0.42, or 42%. So clearly, more often than not, we're going to be pulling out either a homozygous dominant or heterozygote individual. The chance of me reaching in and grabbing 2 recessive alleles in a row, making a homozygous recessive individual, is 0.09, or 9%. All right, so we've taken our allele frequencies, we've plugged them into this formula, and we've figured out how often we would expect to see our different genotypes. So here's just another way of looking at that. Okay. So going back to that assumptions list, if none of those assumptions have been violated, so if this is an extremely large population, if there's no random mating, if there's no mutations, then in the next generation, we should have the same exact allele frequencies. So we should have the same chance of getting an A1 allele and an A2 allele. So if we're measuring one population, we look at the next generation and we compare those numbers. If they have not changed, we can assume that they're at Hardy-Weinberg equilibrium. All right, so we do that calculation again. We get those same numbers because they're at the same frequency. This population is at Hardy-Weinberg equilibrium. So here's just the extension of that. Because we're putting all of these alleles into that same pool, nothing's changed. We're going to have the same allele frequencies in the next generation. So it's our null hypothesis, because if those allele frequencies do change, then we have evidence that something else is going on. So again, null hypothesis, nothing changes. Hardy-Weinberg Principle looks at populations, and we're assuming that they're under Hardy-Weinberg equilibrium. And all that means is that that population meets those 5 assumptions that we mentioned earlier. So if those frequencies do change over time, then we can start to figure out what would be different. So what evolutionary process is contributing to these changes? So then scientists who are studying this, population geneticists, can go in and say, is it selection? Is it gene flow? Are different populations able to get over this river? Is there a high rate of mutation? Is there non-random mating? So once we know, or once scientists figure out what we would expect under Hardy-Weinberg equilibrium, we compare that to what we actually measure in our population, and if it's different, then we can start to investigate which one might be contributing to why these numbers are different. Okay, so again, we're just using this as a null hypothesis. Most populations on Earth probably don't meet these assumptions. However, sometimes even if the populations don't, certain genes within that population may or may not be under Hardy-Weinberg equilibrium. So we use it as a null hypothesis. Just because populations probably don't meet those assumptions doesn't mean that this idea isn't helpful. Okay, so scientists compare those numbers and figure out what evolutionary process could contribute. So we're going to practice doing some of these problems. So before we get to that, if you've been following along okay, you should be set. But if you want to get a little bit more background, there's a really helpful Khan Academy video. It's about 5 minutes. So go ahead and pause this video, mosey on over to applying the Hardy-Weinberg equation in this Khan Academy video, give that a listen or a watch, and then come back. Okay. So again, if you've been following along okay so far, go ahead and continue. But if you want that extra help, please watch this video. It's really helpful. All right, so if you've watched it and you're back, let's move on. We're going to do some practice problems with these ideas. Okay, so we're scientists. We're studying a population of charmanders. So usually they have this nice orange skin color. Their skin color is determined by one gene, and that gene has two alleles. So we have capital G and lowercase g. So charmanders with the homozygous dominant or the homozygous recessive, homozygous dominant or heterozygous genotype are orange. Charmanders with recessive, homozygous recessive are green. All right, so we've got some rare green charmanders. So we've been studying this population a while. We've done some careful measurements. And we know that 15% of the charmander population are green. So we have a genotype frequency for GG, since this is the only possible way we can get green charmanders. So how do we figure out the allele frequency of this population? We know one genotype frequency. How do we get to allele frequency? Well, we know, for one, our genotype is homozygous recessive, because that's the only way you get green charmanders. So what letter represents the recessive allele that we learned from this lecture? I'm going to grab my pen. So the letter of the recessive, the chance of getting a recessive, is Q. So this is what we want to figure out. We want to know the allele frequencies, so we want to know P and Q. So what term represents the chance of being homozygous recessive? Well, we looked at those three terms that represented genotype frequencies. That was P squared, 2PQ, and Q squared. So specifically, we're looking at the chance of being a homozygous recessive individual, a green charmander in this case. That's going to be represented by Q squared. So we know what Q squared is. We know that 15% of the charmanders are green. Oh, so this is a typo. I meant to replace this. I meant to make it 16. So in this case, we measured this population. We went and double-checked our work, and we realized it's actually 16% of charmanders are green. Okay, so moving along, 16% of charmanders are green. We know that Q squared is the chance of getting our recessive phenotype. So Q squared equals 0.16. So Q squared equals 0.16, or 16%. So all we have to do to find the allele frequency of our recessive allele is just take the square root. So we just take the square root of 0.16, and we can find our Q value. Okay, so we take the square root of 0.16, and we get 0.4. Okay, so we found one allele frequency, 0.4. All we did was take our measurement of green charmanders we took the square root of this measurement to find the allele frequency. So we know Q, so we know how often we should expect to see a recessive allele in this gene pool, but what about our chance of seeing a dominant allele? Well, we know that P plus Q equals 1. So if Q equals 0.4, so P plus 0.4, equals 1. All we have to do is take 1 minus 0.4, and we can say that P equals 0.6. You can probably tell by the clicking that I'm doing this on my MacBook. Okay, so we found Q because we knew how often we saw this genotype. So green Charmanders, only one way to get green Charmanders, so we knew the genotype. The chance of getting GG is represented by Q squared. We took the square root of Q to find P. Sorry, we took the square root of Q to find Q, right here. Because we know P plus Q equals 1, we can say that P equals 0.6. So we've gone all the way from knowing how often we see green Charmanders to knowing their allele frequencies. So because we know how often we should see P and Q, we can use that information then to look at the next generation of Charmanders to see if the allele frequency for P is also 0.6. And if it's not, then we can investigate why that would change. Okay, so hopefully you were able to follow along. If not, I'm happy to work on some more problems. Check out that Khan Academy video. They also go through some of these. That is our lecture on Hardy-Weinberg. We defined a gene pool. We looked at the assumptions that we make if a population is under Hardy-Weinberg equilibrium. You know that scientists use this as a null hypothesis, and here is how we can figure out allele frequencies and genotype frequencies. So